So now we talk about transcription. So as we have already said before, is that the central dogma was basically the flow of information from DNA to mRNA to proteins, right? The procedures through which they pass from DNA to mRNA, it's called transcription. And from mRNA to protein, it's called as translation. Right? So before getting into transcription, we are first going to look at different types of RNA. There are three major different types of RNA: tRNA, rRNA, and mRNA. We start with the most basic, it's the rRNA. The rRNA is basically a ribosomal RNA. Ribosomal RNA. Right? And there are different types. It could be 23S, 16S, 5S in prokaryotes, so 28, 18, 5.8, or 5S. S means a Swedberg unit. And it's basically they centrifuge it. Swedberg centrifuged these molecules, and their centrifugation unit is Swedberg unit, in which they sedimented, the rate of sedimentation, right? So that is essentially present in the ribosomes. And then we have the transfer RNA. It basically has two models, but right here I have made you the clover leaf model, right? We're just going to do clover leaf model because that's what is important. And we will actually detail transfer RNA in the later video when we talk about translation. But right here you see there are loops. So this is the this is the thymine pseudouracil. So this sign means pseudouracil, and this is T, and then cytosine loop. This right here is the variable loop. Then this is the anticodon loop. We will detail anticodon later on. And then we have a D loop, and then here it is the 3' end CCA and the 5' end with complementary base pairings here, here, and here. So that's how the tRNA is structured. tRNA, it is going to recruit amino acids to the ribosome, okay, with the help of the ribosomal RNA. Okay, we are going to detail the above two in translation. Here we have the mRNA. So we need to do a bit of detail here, not much, just a bit of details here. Okay. Right here what you see is mRNA is essentially one long strand, right? It is one long strand of DNA. And here we have a poly A tail at the end. So this is the poly A tail. Then there is a small coding region which actually has information. This has all the information that you need, that is transcripted from the DNA. We'll see how it's transcripted. And then we have a 5', this is the 5' end, and this is the 3' end. At 3' end we have the poly A tail, at the 5' end we have a cap. We will detail how this 5' cap and 3' poly A tail is created. And then this little area right here, this is the 5' untranslated leader sequence. It leads the coding region. That's why we call it as the leader sequence. And after the coding region, there is short sequence, which is called as the 3' untranslated trailing sequence, which means it means nothing. It won't be translated into proteins. Only the coding region is going to do that. right? All of these things are actually done in order to protect the DNA coding region so that important information is not lost. As you know that this mRNA has to travel into cytosol, right? And in cytosol there are going to be a lot of nucleases, there's going to be a lot of DNases and a lot of RNases that might attack it. So this gap, this poly A tail, this untranslated region actually protects the coding region from getting damaged. Okay. Now we need to discuss a little bit about RNA polymerase before getting into the steps of transcription. So RNA polymerase is multi-subunit. It has different subunits. So the subunits, the core enzyme. So first we have the core enzyme. It has 5 subunits, right? 1, 2, 3, 4, and we have 2 alpha, 1 omega, 1 beta prime, and 1 beta. So that's how there are 5 subunits. So we have 2 alpha, 1 omega, 1 beta, and 1 beta prime. Alpha and omega works with enzyme assembly. They are going to assemble all these subunits to work together. Beta prime is going to do template binding. It means it is going to bind on the DNA that needs to be transcripted, right? And beta is the main stuff. It is 5' to 3' polymerase. It is going to basically transcribe the information. But still something is missing. This coenzyme cannot do anything on its own. It's not specific. It cannot recognize where to start. It is essentially blind. So it needs a walking stick. That walking stick is actually a holoenzyme. That is the sigma subunit. It recognizes the promoter region. Okay, that is so cool. We have done that. Now, before we move on, I have to explain what is sense strand and what is antisense strand. So we know that the DNA is double-stranded, like that. Now only one of these strands are actually going to get transcribed, right? So let me change a pen color. Right here, this is the mRNA. That mRNA is complementary to this above strand. Let's say that this is 3' and 5'. So this 3' end strand is complementary to mRNA. So we call the strand that is complementary, that is called as the antisense strand. And this strand, the 5' end strand, it is going to be somewhat identical to the mRNA except the uracils, right? That is going to be identical. That is called as the sense strand. It makes sense, right? So antisense is actually complementary and sense is identical to the mRNA. That is the information that is going forward. Okay, now we go on towards phases. The first phase is the initiation phase. So in initiation phase, we need to locate the promoter regions. The promoter regions are located, there are 2 regions. I will now use this diagram right here. So this region, this is at -35 sequence is TTGACA. And then we have the Pribno box right here. Now, before going, I'll tell you one more thing. What are these numbers, right? You will ask, what are these numbers? I'll explain to you these numbers. These numbers, now, this is the DNA strand right here. And let's say transcription is starting from here. This is where the transcription starts, right? The scientists have called or numbered it +1. So this region is numbered +1. No region is designated as 0. So there is no number 0. There is only number +1. And then everything before the starting of the transcription is in negative numbers. We call these negative integers. All of these are going to be negative integers, and this is called as upstream of the coding DNA, or the coding region, or the transcription region is a better word actually. Transcription region. That is important to know. All of those are going to be negative, and it's called as upstream. Upstream, and it's going to come in later quite a lot. And everything after it is going to be in positive integers, and it is called as downstream regions, right? So now that we know that, what does this integer means? It is just way of charting or naming. These are just like roads, and we have named those roads. These are railroads, and we have named those railroads with numbers. Okay, now at -10 number, like this is +1, starting of transcription starts here, but before -10, this is the Pribnon box, TATAAT, right? So this is the sequence. That promoter region, that's the promoter region. This is going to be recognized by the holoenzyme. This is called as Pribno box, right? And then at -35, we have another sequence, TTGACA, right? TTGACA. This is another promoter region. Here, holoenzyme is going to recognize these two regions. When it recognizes these two regions, the coenzyme is going to get attached here, right? Now I am moving onwards to elongation. Here, the promoter region is going to get, coenzyme is going to get attached to Pribno box, either Pribno box or either -35 sequence, right? It's going to get attached. When it attaches, that activates the RNA polymerase. And then the transcription starts, right? Now, important thing is that when transcription is happening, if I'll just make it right here, there you have your good old RNA, okay, not here. Here, this is the Pribno box. Here is your RNA pol 3 that is going to come here and get attached. Wait, is it? Yeah. RNA polymerase is going to come here and get attached. Okay. And then several short pieces of 5 to 2 nucleotides, they're going to get transcribed into mRNA, but they're discarded. Only when more than or approximately 10 nucleotides are added, then elongation begins. Now, when does elongation begins? When 10 are done, right? So this is the 10th base right here. When 10 nucleotides are transcribed, this sigma factor is going to leave RNA polymerase 3, and it will free the RNA polymerase. Now it won't be attached to the promoter. It is now free. Now it can slide beyond it, and that's how elongation occurs, right? So before it was attached. It was attached, right, like this. This is DNA. This is sigma factor. This is the RNA pol. This was attached. But when it leaves, after 10 bases have been transcribed, now it is not here. It is gone. The RNA polymerase is free. It will run. It will slide like a sliding clamp, right? It is also a sliding clamp. But the thing is that it cannot proofread. Right? It cannot proofread. So in case of any mistake, the RNA polymerase, if RNA polymerase is here, RNA polymerase is here, it is going to stop, and this is a mistake right here, and we have a mistake right here. The RNA polymerase is going to stop here, it is going to backtrack, it is going to cleave this area, and then it is going to correct it and then move on backwards. So it reverses, it will go in a reverse gear, and then it will correct it, and then it will take its car to a forward gear and then move forward. It is unlike DNA polymerase 3. It does not have a proofreading mechanism. Then we have the termination. Now, termination is basically two types. There are two types of terminations in prokaryotes. One is intrinsic or row independent, and the other is row dependent. So in row independent or in the intrinsic, there is a self-complementary sequence is generated, which means that in the DNA, at the end of the DNA, there is some kind of sequence that is C, 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 and then G, 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 G. And that is here, translated by the mRNA. And when this frees itself, it is now self-complementary. Now here is G, 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 and then we have C, C, C. So what happens that this is complementary to this is complementary to this, and this is complementary to this. So it will loop, form a loop. This is how, see, this is complementary, like this. This is complementary, right? This is complementary, and that's why they may form base pairs. And that's how a hairpin loop is formed. When this hairpin loop is formed, it is basically the binding is weak, and it can be easily separated from the DNA. That's row independent. But what is row dependent? What is row? Row is actually a hexameric ATPase with helicase activity. So what it's going to do, it's going to bind to a C-rich ROT site at 5' end. The row is a protein which will join, this is a 5', will join at C-rich protein, site, which is called as the ROT site, right? And then it will move along. This is the row protein, this is the ROT site. This will move along, and in front of it we have the RNA polymerase 3, and it is going to come along here, and it is then going to hit RNA polymerase 3 off of the DNA, and that's how transcription is stopped. And because it also has a helicase activity, so as you know that this transcription is because of base pairing, right? The DNA RNA hybrid is now going to break because of the helicase activity of the row protein. Next, we are going to do this in the next video because I don't want to make a video more than 20 minutes. Thank you so much. See you in the next video.